going on? Riffy here, getting ready to start your nervous system neural communication notes. So if you're going in sequential order, this is going to be part 3B of our nervous system notes. You'll have to excuse me a little bit. Got a cold during this recording, so you just have to bear with me a little bit. But here we go. Alright, so neural communication as it fits in with all of the functions of the nervous system is basically the primary function is to coordinate voluntary and involuntary responses to stimuli or to coordinate and integrate sensory information. So one of the big things that occurs with, as you know through our notes the last couple days, is through action potentials. These are also called nerve impulses. Now, as a nerve sends an impulse down the entire, as a neuron sends an impulse or the action potential down its entire length, eventually it's going to reach an endpoint. That endpoint is going to be like your synaptic bulbs or your axon terminals or things like that, but regardless of what occurs, that neural tissue has to meet up with, at some point in time, some other kind of tissue. This can be another kind of neuron, another neuron. It could be anything from muscle tissue to glands to all kinds of other things. But regardless of what happens, there has to be some kind of communication that occurs between that neuron, where the signal's coming from, and whatever tissue's coming up next, because it can't just simply hand it over to it. It's not quite that simple. So what actually occurs is information is transferred from a neuron to another kind of tissue through the use of neurotransmitters. These are those chemical messengers that actually are used to transmit signals from a neuron to another kind of tissue. There's various kinds. There's excitatory and inhibitory, but regardless of what kind of neurotransmitter crosses, that site where a neuron meets up with some kind of other type of tissue are typically called neuroeffector junctions. You'll also hear these called neuromuscular junctions if it's with neurons, but a generalized term is just a neuroeffector junction. Okay. Like I said earlier, one of the biggest examples which we've already studied is what occurs between a neuron and muscle fibers when communication to contract actually needs to be sent between the neurons and the actual muscle fibers themselves, which is essentially the same process, but we're more so going to concentrate on the function of the synaptic bulb or the presynaptic neuron. So let's rewind and go over what a synapse structure consists of. So at the very end of any kind of axon terminal is going to be what's called the synaptic knob. It's called the synaptic knob because it looks like the knob of a door handle, and it's actually the very end where the synapse occurs. The signal that travels down an axon will reach the synaptic terminal and then the synaptic knob of what is called the presynaptic neuron. Now, if the communication is occurring between two neurons, so the signal has to go from one neuron to another, for example, where a sensory neuron would synapse with an interneuron, or where an interneuron might synapse with a motor neuron, or even possibly where a motor neuron might synapse with other motor neurons, if it's a divergent pool or a convergent pool, it just depends. But wherever the signal is coming from is the presynaptic neuron, and where it's going to is going to be called the postsynaptic neuron. Now, connections between neurons can occur at a lot of different places. Most of the time they occur at dendrites, but they can also occur at cell bodies and at axons. And it doesn't actually matter where that connection occurs. It doesn't have to be necessarily at the very ends. That space that occurs between the presynaptic and the postsynaptic neuron is called the synaptic cleft, but you already knew that from back when we talked about neuromuscular junctions. I like this diagram. It looks like a lot is going on here because it kind of does have a lot of information in it, but one thing that I really like that it shows, and you'll notice, is that it shows where synapses can occur at various locations. So you see here that there's a neuron synapsing with the soma or cell body, but that same neuron is also synapsing with the dendrites. And then you'll see down here that you have the axon terminals synapsing with the dendrites of another neuron, and you have various other synaptic terminals synapsing with other dendrites. It's very nice. It shows everything that really goes on here, and it also gives you all the different properties of an individual neuron itself. So other typical structure things that are going to occur at a synapse is you're going to have at the synaptic knob, you're going to have vesicles. If you kind of go back to your biology days, vesicles are essentially packets of some kind of product. So a neuron, one of the things it produces or creates inside of itself is actually going to be the neurotransmitter that is used to communicate with the other types of tissue. Those neurotransmitters, once they're created, get packed in vesicles where they kind of just chill, like it's the inside of a FedEx truck, until they actually have to be delivered. It's a big key player in this. There are many different kinds of neurotransmitters, like we talked about a little bit earlier, but it basically comes down to how they're going to function in terms of transmitting a signal. You can have inhibitory or you can have excitatory neurotransmitters. They're basically mean exactly what they are. Inhibitory neurotransmitters essentially depress an action potential. So their main function is when they get to the very end and they're secreted in that synaptic cleft, they try to keep any signals from occurring outside of that particular neuron. Then there's also excitatory neurotransmitters, which they cause rapid depolarization and they promote action potentials, meaning they try to get more and more action potentials to go. So what I was saying is, if this is going to be the synapse, this is going to be this little section right here, all the way down to here. This is your presynaptic neuron, and anything afterward is going to be postsynaptic. So let's just pretend for all practical purposes that in this two neurons synapsing, what you're going to end up with then are a couple different key features here. So what you have is you have your axon terminal, and then that axon terminal has within it different things. Since it has to go through things like some kind of active transport, particularly with these vesicles, which you're going to find in a second here, where they go through some kind of exocytosis, they're going to require ATP or energy, and that's where the mitochondria are going to come into play. You have your presynaptic neuron, which encompasses this entire section right here. Then you're going to have your postsynaptic neuron, which is going to be anything afterwards. In that area between the two is going to be considered your synaptic cleft. Now, all of these orb-like structures here at the bottom are going to be your synaptic vesicles that contain the neurotransmitters. What they're demonstrating down here is the neurotransmitters being expelled into the synaptic cleft, which we're going to get into here in just a few minutes. Alright, so examples of how synaptic functions with neurotransmitters. Essentially, you're going to get some very fantastic, riffy animations here in just a second, but the first thing that happens is all of that action potential generation and propagation stuff that we've been studying the last 2 days travels down the entire length, starting from the dendrites, going through the somatic cell body, axon hillock, all the way down the axon, until it reaches the synaptic terminals, where it then travels all the way down to the presynaptic knob, or presynaptic terminal, or presynaptic neuron, whatever you want to call it. What that then causes is, on that presynaptic neuron, there's going to be calcium channels, and outside the neuron are going to be calcium ions. That action potential, or that influx of sodium that travels all the way down, because you know that as an action potential propagates down a neuron, sodium rushes in, depolarizing, and potassium rushes out, and that continues all the way down. Well, that influx of sodium triggers calcium triggers calcium channels to open up and allow calcium from outside of the neuron to enter the neuron. So here's where we get into our unbelievably amazing riffy animations. You have your presynaptic neuron all right here. These pink channels right here that are closed off, those are going to be our calcium channels. Here you have your synaptic vesicles. The stars are going to be our neurotransmitters. Here's your synaptic cleft. The green is going to represent sodium ions, and then the green channels down here in the postsynaptic neuron are going to represent sodium channels. So let's discuss what happens here. So as that neurotransmitter, as the action potential travels down to the ends of the synaptic terminals, to the synaptic bulbs, that causes, going back again, that causes the calcium channels to open. Once they're open, that calcium ion is going to rush inside the synaptic bulb. Now, this calcium acts as a trigger for the next step in the entire reaction. Once that calcium actually rushes in, it causes synaptic vesicles with their neurotransmitters inside of them to move down to the synaptic bulb and actually fuse with the membrane of the nerve cell, or you could call it the neurolemma. What then occurs is any neurotransmitter that's inside of that synaptic vesicle, it uses active transport to exocytosis to expel all of the neurotransmitters into the synaptic cleft. So that calcium that is rushed in is going to cause the synaptic vesicle to move down to the synaptic bulb, which it's going to fuse with, and then expel the neurotransmitter into that synaptic cleft. Now, nothing is occurring until that particular neurotransmitter moves through diffusion across the synaptic cleft and actually binds with what are called postsynaptic membrane receptors. These postsynaptic membrane receptors in this particular instance are going to be those that are going to tell sodium ion channels to open up. When that neurotransmitter binds with these specific sites on that postsynaptic membrane, it causes the sodium channels to open up, which allows sodium to rush into the inside, which then starts all over that action potential generation and propagation that occurred way at the beginning in the dendrites, soma, or cell body, moving on to the axon hillock. So we pick up where we left off again, where we have our neurotransmitters that were expelled by the synaptic vesicle, the stars. What they're going to do is they're actually going to move to specific sites which cause those sodium ion channels to open up. Now, if you notice correctly, or if you notice that actually the neurotransmitters are still located or attached to the specific sites that have caused those to open. So let's look at this again. They diffuse across the specific sites, cause the sodium ion channels to open up, and those sodium ions rush in. Keep in mind again, those neurotransmitters are still right there. This is going to play an important part here in a second, because you will continue to send a signal, or neurotransmitters will continually be released until those sodium ion channels are closed. In order to close those sodium ion channels, you actually have to first close the calcium channels from the presynaptic neuron, which stops the neurotransmitters from leaving the synaptic vesicles into the synaptic cleft, which then will also then stop them from diffusing across and meeting up with the postsynaptic receptor sites. Once that occurs, that neurotransmitter is actually broken down through enzyme reactions, which breaks down into simpler substances, and any neurotransmitter that actually wasn't used get actually reabsorbed back into that presynaptic neuron. Any neurotransmitter that was used can be broken down into simpler substances, which are then also transferred back into the presynaptic neuron and are reassembled back into neurotransmitters. So it's really nice that some of those neurotransmitters is almost a recycling center type action that occurs with any of the neurotransmitters in the synaptic cleft. So if you look at it here, what we're going to show is pay attention to the neurotransmitters at their specific sites, these stars down here. They're going to slowly start to be dissolved through that action of enzymes within the synaptic cleft. If you remember correctly, back when we talked about the neuromuscular junction, the enzyme that actually broke down acetylcholine, which is a neurotransmitter, was acetylcholine esterase. What I don't have animated here is once that's broken down, it would be reabsorbed back into synaptic vesicles and reprocessed and created into fully functional neurotransmitters, or if there were extra neurotransmitters located in this synaptic cleft, they would be reabsorbed and actually packaged back up into vesicles. What I have here for you are some pictures, I believe actually straight from your book, that kind of go through this process, and they have a little bit more. I mean, there's no animation, there's no fantastic riffy animations here, but it goes through all of the steps. So what you end up with is in the presynaptic neuron, you have the action potential that reaches this particular point in the synaptic bulb. All that influx of sodium causes calcium ions, which are down here, to rush in through calcium channels. This influx of calcium ions causes synaptic vesicles with a neurotransmitter, here it happens to be acetylcholine, to fuse with the cell membrane and expel their neurotransmitter into the synaptic cleft. That neurotransmitter then diffuses across the synaptic cleft and attaches to specific receptor sites on the postsynaptic membrane. Once they attach there, it causes the channels to open up, allowing an influx of sodium to rush into the cell. Once that influx of sodium carries in there, you get the propagation of an action potential in the next particular type of tissue, whether it be another neuron or muscle tissue. Something that's important to understand here, if you notice, it is being broken down and it is about to be reabsorbed back into the actual presynaptic neuron. Right here, you can tell they are being packaged into vesicles, which will eventually be used. Let's watch this YouTube video real quick. Maybe. Possibly. Please. Or maybe it just doesn't want to work. Oh, fantastic. That's exactly what I wanted to see in the middle of my lecture. So here's what I'll do. These two YouTube videos, I'll post those links at the very end. So where does this lead us after this? Also, sorry, right here. This is a web tutorial where you can practice and click through animated steps for all of this process of neuron to neuron communication. But so what happens when we need this signal to travel from a neuron to other places? This is where those neural pools come into play. So I'm kind of going to hit this fast considering we did an entire lab over it. But there's two kinds of neural pools. You can have coming from one source and spreading to multiple, or you can coming from multiple sources converging onto one. A divergent neural pool is when you want information to spread from one neuron or from one neural pool to several neural pools. It diverges into different areas. Your eyes are one of the best examples here. So a lot of that information that comes in from sight is actually routed to different areas of the brain for things like posture, balance, coordination, sense of knowing where you are in your environment. On top of the visual stimuli, you can process visual information for reading, writing. You can access memory. All of those things, one signal coming into your head, routed to multiple parts of your brain. Other examples are stepping on a nail. So you have that one single input signal that are spread to multiple places. It diverges to multiple. When you step on a nail, that signal that comes out of your brain is going to be spread to multiple types of muscles in order to coordinate the balance involved in lifting your foot, standing on one foot, and trying to remove a nail from the bottom of your foot. And then you have a convergence. This is where several neurons synapse onto a single postsynaptic neuron. What this does, one of the biggest ones, is that voluntary and involuntary control of your diaphragm. For example, if you wanted to breathe, I could tell you to take a nice deep breath. Well, that is coming from a conscious control center of your brain, which that signal converges onto the diaphragm. But you also have that subconscious control of respiratory that also converges into one point on the diaphragm, which is a type of muscle tissue that you can consciously control or subconsciously control. And that's the end of the video. Like always, if you have any questions, I thought this was kind of a review considering we hit it so hard in the neuromuscular junction section and muscular contraction. But as always, if you have any questions, please let me know.